All right, membrane function, and we're really going to focus in this chapter on transport. There are other topics that are covered in this particular chapter on membrane function, but again, we are going to be focusing on the transport aspect. So let's take a look here at the different types of transport. We have something called passive transport, active transport, and then we have this bulk movement kind of transport that can occur. So passive, this requires no energy, no energy. And the most common, which you might be familiar with, form of this transport is diffusion. So something moves from an area of high concentration to an area of low concentration. So for instance, you drop your favorite perfume in the front of your classroom, let's say. It's a travesty, but it happens. Now, will the person in the back of the room, the very back of the room, will they smell that perfume immediately? No, they will not. Will they eventually smell it? Oh, yes, they will. Because those molecules are going to diffuse from the area of high concentration, which is where the perfume bottle has broken, all the way throughout the entire room, requiring no energy at all, moving from that high concentration to the low concentration in every corner of the room, and it's going to distribute evenly throughout the room over time. Okay, so that's diffusion. Now also osmosis, this is another type of passive passive transport, and it is specifically the movement of water across a membrane. Now water loves the solute. I can't tell you how much it loves the solute. Water is like the biggest fan of the solute. Let's just say that water is a fan of, let's just say, your favorite music band. And it goes to the concert and it is just crazy about these solutes, which would be the band members. And it just wants to go. Whenever it sees those solutes, the band members in this example, it wants to go to them. It wants to go to them. All right, and then we have something called facilitated, and that is movement of solutes or particles, and sometimes water, through a protein channel in the membrane. Now again, it doesn't require any energy, it's just the molecule is moving through the actual membrane through a transport protein. All right, and then we have the active transport, and that is going from an area of low concentration to an area of high concentration. So that would be like putting all of those solute particles from the perfume that have diffused through the air back into the bottle. That would be very difficult and require a lot of energy. An example would be the sodium-potassium pump, and that example is described in your textbook, and I believe there are some questions about it in the study guide. And then finally, bulk transport, that really is the bulk movement of very large molecules either into the cell. Endo, entering, is into the cell, and then exo means that large amount of material is exiting the cell. And there is actually a nice animation showing you both endo and exocytosis on Isidore. So please take a look, and then also know the difference between phagocytosis, pinocytosis, and then something called receptor-mediated endocytosis, all types of endocytosis. All right, so let's move on to our next slide here, and we're going to talk about how solutes and water move through our cells or in and out of our cells. And we first have to talk about the tonicity of a cell. Now, whenever you're talking about the tonicity, how concentrated something is, you're always talking about the cell in relation to the fluid that surrounds the cell. So for instance, here we have, let's say, a solute concentration in the cell of 5. And the fluid outside the cell in blue would be a concentration of 15. Now, there are fewer solute particles in the cell, so it is called low, hypotonic to the solution. You don't the convention is not to give the solution outside the cell a tonicity name. You only talk about hypertonic, hypotonic, or isotonic when you're talking about the cell. And the cell is, for instance here, again, hypotonic to the solution. Let's look at another example. Oh, now we have a cell that has a solute concentration or solute particles that are higher than the solution. So these solute particles would be hypertonic, or the cell would be hypertonic to the solution. And something I forgot to mention, let's go back here. How would water move? Remember, water loves the solute. Where is there more solute? There is more solute out here. So water would move out of the cell, okay, in this situation. And the cell would shrink or shrivel. In this situation, water would move into the cell. Now, if there's a very large difference, if you'd have a lot of water moving in, this cell, being as though it looks like it's just a cell of an animal, might burst as a result if too much water moves in. All right, now isotonic, it would just mean, for instance, we'd have 20 and 20. There would be movement of water, but there'd be no net movement of water. Okay, so just keep that in mind. And then finally, we have an example here, and there are several examples, and there's practice sheets posted on Isadore for you to take a look at and work through these problems of having a cell and then the environment, which would be the solution outside the cell. And I give you certain sugars, for instance, here, and then I tell you what the membrane is permeable to. That's very important. So our membrane here, it's permeable to water, which you can assume that, but I will tell you that because most membranes, all membranes, permeable to water if it's a living cell. And the simple sugar glucose, so glucose can move across, and so can sucrose, but oh no, not fructose. So the thing you want to do here is you want to figure out, well, how much glucose do we have in the cell? How much do we have outside the cell? We have a total of 11 glucose molecules. And those can move across. So they're going to even out over time. So I just put this cell into this beaker. That's the idea. So 6 and 5, that's 11. So at the end of the experiment, when everything comes to equilibrium, we'll have 5.5 outside, 5.5 inside. And then the same with sucrose, 1 and 4, that gives us a total of 5. So 2.5 outside, 2.5 inside. Now fructose, remember, that doesn't move across. So at the end of the experiment, no matter how long you wait, you'll have 2 inside, 3 outside. And then you'd add those up, those solutes, the glucose, the fructose, and the sucrose at the end of the experiment. And depending on which one had a higher number of solute particles, you would say, or you could determine how water would move. Would there be a net movement of water? And you can assign a tonicity to the cell. All right, so that covers the major ideas or topics for membrane transport and membrane function.